Hello and welcome to Bio40 Pre-Lecture 17. Today we're going to discuss the genetic code. So let's get right to it. This is it. This is the genetic code. These are the rules for turning a sequence of bases into a sequence of amino acids. This table shows the code using RNA bases, but the same table would apply to DNA if you just replaced the U's with T's. 3 nucleotides correspond to an amino acid. For instance, you can see here that the sequence UGG corresponds to the amino acid tryptophan. We call these 3 nucleotide groups codons, and they're often likened to the words of the DNA language. Having 3 nucleotides per codon makes sense, since this is a minimal number it would take to assign a different codon for each amino acid. If codons were only 2 bases long, there would only be 16 possible combinations, not enough for the 20 amino acids. However, 3 nucleotide codons allow for 64 possible arrangements, far more than are necessary. As a result, the code is degenerate, which means there can be multiple codons for each amino acid. Some amino acids only have a single codon, such as tryptophan and methionine here, but if we look at serine, we see 6 possible codons. We also say the code is unambiguous. That is, a given codon will always correspond to the same amino acid. ACC is always threonine and not anything else. CGG is always arginine, and so on. The code is non-overlapping, which makes the reading frame particularly important. To make sure the gene is read in the right frame, genes always begin with the methionine codon AUG, which is also called the start codon. The sequence ends with the stop codon, a codon that doesn't correspond to any amino acid. These are UAA, UAG, and UGA. Finally, it's important to note that this table is nearly universal. Genetic information is read the same way in E. coli as it's read in humans. There are a few exceptions out there, but they only have very minor variations from the standard codon table. There's no reason that the codon table needs to be arranged in this particular way, so this is good evidence of a single origin of life. Let's go into a little more detail about reading frame issues. Say this is a stretch of DNA that contains a gene. Most genes actually have dozens to hundreds or even thousands of codons, but this will do for our example. Reading the strand in the 5' to 3' direction, our first start codon, ATG, is here. So the first amino acid is methionine, as it always is. With the non-overlapping code, the next codon will be the 3 bases that follow, GGA, which is glycine. Next is CGG, which is proline. And so on, until we get to this codon, TAG, which is a stop codon, marking the end of the amino acid chain. But the code didn't necessarily have to work this way. An overlapping code would be possible. Let's imagine what that would look like. We still have ATG as a start codon, so we still begin with methionine, but now we just shift 1 base over. So the first 2 bases of the next codon are the last 2 bases of the previous codon. Here, this is TGG, which is tryptophan. Then comes GGG, which is glycine. Then GGA, another glycine. And so on, down the gene. Though we actually have a different endpoint this time. This TGA sequence didn't end the amino acid chain in the previous method because it was not in the reading frame. But with overlapping codons, it will be read as a stop codon. You could imagine some potential benefits to an overlapping system. It's certainly more economical. A given stretch of DNA could contain 3 times as much information with overlapping codons. Also, single nucleotide insertions and deletions would cause less of an impact. Rather than causing a frame shift, they would just result in the addition or subtraction of a single amino acid. But there is a very major drawback. Overlapping codons would severely limit the number of potential amino acid chains that could be made. With ATG as a start codon, the second codon would always have to be TG something. There are only 4 different codons that could be in that second position. Likewise, once the second codon is decided, there are only 4 possibilities for the third one, and so on down the line. Such a limitation is just too great, so the non-overlapping system is used. We've talked about frame shifts a couple times already, but I want to go back to it now that we know the genetic code. Let's continue with the sample gene we were just looking at and insert a single adenine between these 2 bases. So this becomes our new sequence. All the codons before the insertion are unchanged, but the codon at the site of the insertion has been altered from a proline to a histidine. And not only that, every codon that follows is different. You can also see that a stop codon that wasn't in frame before now is in frame, so the amino acid chain is shorter than it should be. The other types of mutations we talked about before should make more sense now too. Let's look at a substitution where this adenine has been replaced with a thymine. Instead of being AAG for lysine, there's now a TAG, which is a stop codon. This is a nonsense mutation. Truncating a protein halfway in is very likely to mess up its function. Now let's imagine the same base was changed to a G instead. GAG still codes for an amino acid, so this is a missense mutation. No amino acid before or after the altered codon is affected, though this still could be a significant change. We've gone from the positively charged lysine to the negatively charged glutamate. If the amino acid in this position plays a key structural or enzymatic role for the protein, such a change could greatly harm the protein's function. But if the amino acid in this position isn't so important, the change may be tolerated and have no phenotype at all. It's highly situation dependent. Now let's say it was the next base over that was replaced with a guanine. This changes the amino acid from a lysine to an arginine, another missense mutation. But since lysine and arginine have similar chemical properties, both being positively charged, we call this a neutral mutation. Such a mutation could still damage protein function, but since the amino acids are similar, this is less likely. Finally, let's imagine the third base in this codon is changed from a guanine to an adenine. Well, both AAG and AAA are lysine codons, so this mutation hasn't altered the amino acid sequence at all, and there should be no effect. This is a silent mutation. So these are the major things you need to know about how the genetic code is read. But a major theme for this course is that it's not enough to just be aware of how something works. You should be curious about how we know these things are true. So we're going to spend the rest of this lecture, and the next one, going over the experiments that allowed us to figure out these rules. First, we return once again to Francis Crick. Crick was at the center of many of the great discoveries in molecular biology. We already talked about his role in determining the structure of DNA, and how he coined the central dogma of molecular biology. Now we're going to discuss how he helped figure out the rules of the genetic code, and this won't even be the last time he comes up. While I don't want to overemphasize the contributions of any one person, after all, determining the structure of DNA would not have been possible without contributions by Watson, Franklin, Wilkins, Chargaff, and others. Crick was certainly one of the most accomplished scientists of his era. His work on the genetic code was published in 1961, and this too was the culmination of work by several great scientists, including Sidney Brenner, who would later go on to win a Nobel Prize of his own. They used the bacteriophage T4R2 system that we previously discussed. Recall that wild-type phage forms small fuzzy plaques on both E. coli B and K12 lambda strains, while R2 mutants form large plaques with distinct edges in the B strain and can't form plaques at all with K12 lambda. This allows both screens and selections to be carried out. Crick et al. Focused on the R2B gene, inducing mutations with proflavin, which you'll recall is an intercalating agent and leads to frame shifts. Though at the time, DNA sequencing wasn't possible, so Crick et al. Had to infer proflavin caused frame shifts based on their data. Frame shifts are a convenient type of mutation to work with, because their effect is often severe and they are rarely leaky. However, they're still point mutations, so reversions are possible. Crick et al. Mutagenized phage to get point mutations, then mutagenized again and selected for wild-type revertants. Another interesting feature about frame shifts is that revertants are possible without directly reversing a mutation. For instance, a deletion at one spot could be fixed by a nearby insertion. Look at the example on the right. Here's a simplified sequence, which is just a codon ACG repeating. This corresponds to a chain of threonines. But imagine there's a deletion at the adenine of the second codon. The frame from that point on is read as CGA repeating, a chain of arginines. But if a base is added nearby, the ACG frame is repaired, and the threonine chain continues. Only the codons between the two mutations are altered. As long as the protein can tolerate the changes to these two amino acids, its function will be intact. Think about what sort of codons would be least likely to be tolerated. A true reversion to the wild-type sequence would be called a back mutation. But a mutation at a second spot that restores a wild-type phenotype is called intergenic suppression. So Crick et al. Took a frameshift mutant, mutagenized it again, and collected wild-type revertants. But how could they tell if reversion was due to back mutation or another mutation in the same gene? Well, they co-infected their revertants with true wild-type phage, allowed recombination to occur, and checked to see if they could isolate single mutants. In many cases, they were indeed able to, indicating that the revertants were due to second frame shifts elsewhere in the gene. In this way, they were able to collect about 80 revertants, which were themselves frame shifts. They dubbed their original mutant a +1 shift, and their revertants a -1 shift. With no sequencing, they just had to guess. It would have been completely possible for their original mutation to be a deletion, and the revertants to be insertions. But swapping the signs would not alter their overall results. Next, they made various combinations of mutants to see how they functioned. They saw that a +1 mutation with a -1 mutation would usually be functional, but putting two +1 mutations together would not. Same with two -1 mutations. But 3 +1 mutations, or 3 -1 mutations, would often appear wild-type. Here's a table showing the full pattern they saw. Combining mutations with the same sign would work in multiples of 3, but anything that wasn't a multiple of 3 would still be mutant. This data is consistent with codons being 3 nucleotides long. Inserting 1 or 2 nucleotides would throw off the reading frame, but inserting 3 nucleotides just adds one more full codon. Likewise, 1 and 2 nucleotide deletions mess with the reading frame, but 3 nucleotide deletions just shorten the protein by 1 amino acid. Again, at the time, they couldn't have been sure that what they assigned as a +1 was really an insertion and not a deletion, but this doesn't affect the results. After all, the +1+1+1 combination has the same phenotype as the -1-1-1-1 combination. And an interesting thing to note is that from a reading frame perspective, a single nucleotide insertion is equivalent to a 2 nucleotide deletion, and a single nucleotide deletion is equivalent to a 2 nucleotide insertion. From their results, Crick et al. Were able to draw 3 important conclusions about the genetic code. First, it is a triplet code, for the reasons we just discussed. But they also interpreted their results as evidence that the code is non-overlapping, and that it is degenerate. That is, multiple codons can code for the same amino acid. It may or may not be obvious how their results supported these last 2 conclusions. So first I'm going to see if you can explain it to me, and then we'll go over it in class. That's it for this lecture. Next time we'll discuss the research that allowed us to identify which codons correspond to which amino acids.